Great. Our next and final speaker of the session is Chris Summerfield. So we're pleased to have Chris here. He is a lecturer in experimental psychology at the University of Oxford. He's done a lot of interesting work on understanding human decision-making, in particular with regards to kind of perceptual tasks. So he does have experiments, but also kind of taking that to computational modeling and understanding how the neural basis for action. So welcome, Chris.
Thanks, Percy. Thank you all for being here, and thanks to the organizers for inviting me. So like Percy said, I'm a psychologist and I work on human decision-making, but I also do a bit of moonlighting at Google DeepMind. And one of my interests is how we can learn from biology and use that knowledge from neuroscience or biology to inform our building of artificial agents. So humans intent on building new technology have often looked to biology for inspiration. And I think it's fair to say that the results haven't always been very successful. But of course, recently, engineered solutions to intelligent behavior in complex dynamic environments have been very impressive, just like the work that Hong-Lak was just showing us. So you all know about DQN, which is able to behave intelligently in dynamic adversarial environments with end-to-end training from just pixels. Now, of course, DQN is a model-free system, which uses a convnet to approximate the complex value function in each game. And for me, as a neuroscientist, I'm very surprised by just how well a model-free system can do in an environment like Atari, where there's such strong temporal dependencies between states. But of course, DQN doesn't do well on all of the Atari games, and in particular it tends to fail in those games where rewards are particularly sparse, and so the harvesting of rewards would particularly benefit from having a good model of the world. And of course this follows on nicely from what Hong-Lak was just telling us about. But of course, you as humans, you do have a model of the world and you use it to navigate, like you, or many of you may have done this morning, when you took Barcelona's metro to get here. And so what I'd like to do in my talk today is to try and give you a perspective from neuroscience about how this might be achieved in biological systems. So what's the difference between a neural network like DQN and the human brain? Well, I guess lots of things. But maybe one of the most salient is that, of course, supervised neural networks involve largely undifferentiated layers of units that are trained with a single global optimization principle. And that's very different from biological brains. So biological brains are highly modular, and each region seems to be specialized for a different function, relating to, for example, perception, or attention, or memory, or higher cognitive functions, including language, like Joel was telling us about. So if you take, for example, the hippocampus. So here I'm showing you the hippocampus both on an MRI scan, that's the area in yellow here, and also in this beautiful drawing from Ramon y Cajal. So the hippocampus has highly stereotyped internal circuitry, and there's extensive evidence that suggests that it uses this circuitry to carry out very specific computations that underlie particular forms of learning and memory. The hippocampus is also, of course, connected with the neocortex in a very specific fashion. It shares connections with some regions and not others, and it's particularly densely interconnected with these medial prefrontal regions, which I'll talk a little bit about later. So recently at DeepMind, many of you will know that we showed that by adding a modular storage system, perhaps a little bit like the hippocampus, to a standard neural network, we can get the network to perform well on a whole bunch of memory and reasoning tasks, including that problem of how to navigate in the subway. So the differentiable neural computer uses a standard recurrent neural network, but rather than directly controlling the outputs, the RNN, of course, controls what is written to or read from a content-addressable memory. And this allows the network to perform tasks such as computing the shortest path in a graph, like the London Underground system, without any built-in prior knowledge. So on each training cycle, the network receives symbolic inputs that describe the graph as a series of nodes and edges, and it's then provided with a start station like Moorgate and a destination station, Piccadilly Circus, and a query, and it has to spit out the shortest path between these as a series of nodes and edges. And unlike other architectures, for example LSTMs, it's able to do this accurately with fully end-to-end training. So that's very impressive, but I would argue that we are still some way from building neural networks that can solve navigation and planning problems like biological systems do. So we've known for many years that, never mind humans, even the humble lab rat can learn a generative model of the world, what psychologists call a cognitive map, and use it to plan in a maze. So let me describe a classic experiment that was done more than half a century ago now by Tolman. So there were two groups of rats, and these rats were placed in a maze which had a food box at one exit. So one of the groups of rats, for the first 11 days of training, received a reward whenever it reached the food box. And the other group was simply taken out of the maze with no reward. So then on the subsequent days of training 12 to 17, both groups were rewarded, and the dependent measure was how well the two groups did. And so what's interesting is that the group who received no feedback during the initial training, so those are essentially the group that's receiving no supervision, did far better on the later training phase than the group that received supervision did. So in other words, the rats are learning this cognitive map without supervision, and they're then able to apply it to rapidly solve a new problem. So how do rats do this? Well, to understand how they do, it's helpful to examine the neural coding schemes that characterize brain regions such as the hippocampus, whose integrity is essential for navigation and memory. So many of you will know that the CA3 region of the hippocampus, shown here, this is a slice through rat hippocampus. This diagram shows you where the CA3 is. The CA3 region contains neurons called place cells that fire whenever the animal occupies a given location in the environment, but importantly are invariant to its direction of travel or viewpoint, or largely invariant to its direction of travel. So place cells are obviously excellent candidates for being building blocks of a cognitive map. So, but of course, to be useful, a cognitive map needs to describe the likely transitions between states under a given policy in order to encode their expected future occupancy. So imagine now an agent, just like a rat, making random walks through this open environment here. So if we simulate some place cells along the way, then those place cells will fire in a correlated fashion that depends on their adjacency, right? So that allows us to compute a covariance matrix that describes the covariance among patterns of firing in these place cells. Now, the first principal component of this covariance matrix can, of course, be learnt by a simple neural network that's trained with a Hebbian learning scheme. But the interesting thing is that then if we take that principal component and we back project it onto the environment, then what we find is that that component encodes spatial locations with this kind of hexagonal repeated pattern. So this is a highly efficient representation of the expected future occupancy of different states, so where the rat might be likely to go from any particular state that it's currently occupying, that might be really useful for planning. And of course, as we know, this hexagonal pattern mirrors the spatial tuning of another class of cell, known as grid cells, that are found in an adjacent brain region, a region that's adjacent to the hippocampus, called the entorhinal cortex. So the entorhinal cortex, particularly the medial portion, is densely interconnected with the hippocampus. And you can see here, I'm plotting the tuning profile. So these are a smooth rate map of where a particular cell fires as the rat moves through this open arena here. And you can see place cell over here on the right. So place and grid cells were first discovered in the rodent, and of course, John O'Keefe and his colleagues won the Nobel Prize last year for that discovery. But recent studies have developed methods for measuring aggregate grid cell activity in humans using functional brain imaging. So this slide describes the first study to do so. So humans were placed in a virtual reality environment, and they navigated through an open arena, and just like the rats. And you can see trajectories that they took here. And what the experimenters, Neil Burgess and Christian Doller, did was to take half of the brain imaging data recorded in the scanner and to fit a quadrature filter to it with a period of 60 degrees. And the logic of doing so is that this filter will capture, because it has an angle of 60 degrees, it will capture activity which is going to be greatest as the humans make trajectories which fall along these kind of hexagonally aligned grid cell axes in the entorhinal cortex. And so having used half of the data to estimate the orientation, or the putative orientation, of the grid in the human navigation system, they then use that data to fit it to the other half and try and predict whether there'd be a sinusoidal periodic modulation of the bold activity, the brain activity, as humans follow trajectories of 60 degrees through the environment. And that's exactly what they found. And so you can see here the firing rates as humans made trajectories which were separated by 60 degrees, i.e. falling just along the major axes of this grid, was greater than for other axes. So, in other words, what this tells us is that both humans and rodents might use this coding scheme to navigate through the environment. And most interestingly, they found this not only in the entorhinal cortex, where where grid cells have been discovered in the rodent, but also in a number of other regions that are connected with the entorhinal cortex, including this medial prefrontal region. So, this suggests that maybe these grid representations might be useful for planning. And a later study tested this directly by asking whether you can see similar grid-like representations not only when people were navigating, but when they were just imagining trajectories through the environment. So this study, also by Christian Doller, trained participants to navigate through this virtual environment, which they call Donderstown. So it's a realistic kind of urban simulated environment. And then they placed them in a central location, or a number of central locations in the environment. And they asked them to imagine navigating to target locations which had a particular angle of orientation with respect to the major axes of the environment. And what they found was that when looking at patterns of brain activity, so multivariate patterns of brain activity in the entorhinal cortex, those patterns were more similar when participants imagined trajectories which were separated by 60 degrees, again, aligned to the major axes of the grid, than when they imagined trajectories that were separated by other angles. So in other words, this suggests that when we're planning a journey to a destination, we may be using this kind of efficient code, for the covariation among spatial locations in order to plan our journey and to work out where we're going. And this is showing just here the location of the entorhinal cortex, and this is an illustration of how they did the experiment. I'm going through quite quickly, but I'm happy to take questions about the methods at the end. Okay, and this isn't related to navigation, but I can't resist telling you about this beautiful study by my colleague, Tim Behrens, which came out earlier this year in Science, in which he suggested that these grid-like representations may be useful not just for navigation, kind of classic navigation tasks, but actually for the representation of even highly conceptual, novel spaces. So what they did is they trained, rather than training participants to navigate around an urban environment, they trained them on this kind of conceptual bird space. And the bird space was characterized by these visual stimuli, these birds, whose neck and legs varied in length. So there's a 2D representation of the bird space, and here are two birds sitting in this particular space. And after extensive training, what they did was they showed participants trajectories, short videos that describe trajectories through this bird space. And those trajectories were traced out by varying the length of the legs and the neck at different rates. So obviously, if they vary at the same rate, that's an angle of 45 degrees, and if they vary at different rates, then you can create different angles through this bird space. And so using these Döhler and Burgess methods with human participants in the scanner, they show the same six-fold modulation in the entorhinal cortex, and also, once again, in these medial prefrontal regions. So this suggests that this kind of six-fold symmetry in neural coding might be a general property of the neural mechanisms by which we encode representational spaces and plan trajectories through them. So these experiments that I've talked about so far explore neural coding in open environments, but of course we evolved to plan and navigate in much more complex environments, those that are strewn with obstacles, for example. So if you calculate the covariance matrix obtained when an agent makes random walks through a more complex environment, like this 4 rooms environment, then the principal eigenvectors encode the structure of the environment in a hierarchical fashion. So the principal eigenvector here encodes, of the transition matrix, or the covariance matrix, divides the environment in half, and then the subsequent eigenvectors encode individual rooms themselves. And so this is obviously a really useful, compact way to encode this environment. And then you get similar results in other mazes, like this hairpin maze here. And neural signals from humans, too, seem to encode the environment in a hierarchical fashion. So this is a really nice study from Matt Botvinnik's lab, done some years ago now. And they trained participants to make random walks through this room-like environment. So each of these is a state, and it's associated with a unique visual stimulus, such as a fractal. And they showed people random walks through these environments, and they measured brain activity. And remarkably, so in these medial prefrontal regions, even though people had never seen the map, there was differential brain activity when people passed through these bottleneck states, so as if they passed through the doorways, suggesting that this region has access to the structure of the map and knows something about its hierarchical organization. It knows where the transitions between these rooms or contexts are. So it brings me back to the subway map. So how do humans navigate in mazes like this? Well, so in a recent study, in my lab, we tested this by training human participants to navigate through a virtual subway map that we created. And after a long training session, they simply navigated by pressing buttons, like north, south, east, or west, to travel between a start station and a destination station, very similar to the problem that the DNC was faced with. And importantly, we never showed them, or during training, during scanning, they never saw the map. So one useful sort of representation of this map would just be flat, so a kind of encoding a list of transitions among states. So for example, when you're navigating from Martin Station to Dublin Station, you might encode a list of the intervening stations, so Madrid Station, Jagger Station, Clinton Station, and so forth. And we did see, like previous studies, neural signals in the hippocampus and ventromedial prefrontal cortex that vary with the overall distance to goal in terms of the number of stations that you had to go through in order to reach your destination. But of course, a more interesting and perhaps more useful way of representing the map is as a hierarchy of stations, nested under lines. So this type of representation is very efficient for planning because the plan can be represented in terms of the lines rather than a list of stations. So when you're representing a flat plan, rather than representing a flat plan, all the agent needs to know is that to make this journey here is that it needs to kind of go to Clinton Station and change trains, right? Just like you might represent when you're on a subway. So interestingly, when we measured brain activity from humans performing this task, we saw bold signals in a different region encoded the distance to goal in terms of the number of lines or number of contexts that remained to be traversed. And this cost of planning, this hierarchical cost of planning, in terms of the number of contexts to be traversed, was also encoded in human decision latencies, so how long it took them to respond, and those two effects were correlated across participants. And moreover, when we used a multivariate decoding technique to measure the similarity or dissimilarity in patterns of neural activity that were evoked at each station, we found that in the same region, activity was more similar within lines than between lines. So it seems to suggest that this brain region is kind of representing the structure of the map in terms of a hierarchy of different contexts or lines, rather than representing kind of each step en route to the destination. So I'm going to stop there, but this is my take-home message. So biological brains have evolved to represent the structure of the world in efficient ways. And I think that the neural coding schemes that we see in humans and other animals may be useful for shedding light on how to build artificial agents that can behave intelligently in complex and dynamic environments. So I'd just like to stop with an acknowledgement. So that's to my PhD student, Jan, who's sitting over there, who did all the work on the subway task and who's enjoying the conference in his hometown. Thank you all for your attention.
Questions for Chris?
Thank you for the fascinating talk. My question is about whether you could speculate about the causal structure of what we're seeing here. Do you think that there are certain aspects of the, let's say, the biological hardware that implements these mechanisms that makes it particularly useful to represent information in this particular way? Or do you think there's something about the 3D structure of our environment that makes this a particular good coding scheme? And I think, depending on the answer, that your insights may give us different directions in how we need to apply this when we develop artificial systems.
Well, so I'm going to give a very unsatisfactory answer, which is that I think that it's both, right? So we know that biological systems are extraordinarily good at learning, just like some of our unsupervised artificial systems. They're extraordinarily good at learning incredibly efficient representations of the environment. And we see that in vision, where, of course, the preponderance of visual coding or coding in sensory systems reflects the natural statistics of natural scenes, just like it would do for an unsupervised neural network. And I think what I tried to talk about is an example of that efficiency of coding, but in a much more kind of extended domain in which we're thinking about maps and structure. And I think that the discovery of grid cells, it's quite remarkable just how the biological systems have essentially evolved to represent map-like structures in just about the most efficient way that you can imagine. And I think that we have a lot to learn from that. Although I don't have any particular insights about how you would specifically implement that in artificial systems, I'll leave that to you guys who all know how to do machine learning, unlike me.
This is perhaps kind of more of a conceptual question, going back to some of the motivation you mentioned earlier. So you talked about how you were a little surprised that the model-free methods worked well at some of the machine learning applications. Do you think that there is sort of a space for the techniques that, let's say, encode internal generative models of the world to interact with model-free techniques in some way? And what's your take on that?
Yeah, so that question would probably take an hour to answer, I guess, and probably be answered by other people better than me. I mean, my view is that the brain is an unsupervised system, right? So sensory systems are learn almost exclusively in an unsupervised way. I mean, throughout infancy, we're exposed to incredibly sparse supervision systems, and yet, even before we ever encounter a language or start to have much more structured supervision, we have, incredibly rich and complex generative models of the world. So, exactly how we can fuse in an artificial system, we can fuse excellent generative models with, the kind of RL systems, model-free RL systems that do so well in playing Space Invaders. That's the $100 million question. I don't have an answer to that, but I hope someone else does.
Thank you.
All right, let's thank Chris again, and that concludes our first session.